According to the United Nations Food and Agricultural Organization, which measures malnutrition, in October 2010 there were 925 million people that were malnourished. This counts for about 13.6% of the 7 billion world population. Of all those affected by poverty and poor nutrition, children suffer the most. Children in these poor conditions are found to suffer from illnesses 160 days per year on average. These malnourished children also count for at least half of 10.9 million children deaths each year. Many of these poor communities live off staple foods. Unfortunately, living off these plant-based foods causes a lack in many micronutrients. The main micronutrient deficiencies are vitamin A, iron, and zinc. The U.S. government and other organizations such as Harvest Plus work today to discover a way to provide poor communities and countries with food that will provide for vitamin A, iron, and zinc deficiencies. A way to do this is through biofortification. Biofortification is the development of micronutrient-dense staple crops using the best traditional breeding practices and modern biotechnology. Today there are three main types of biofortification being researched and used: fertilization, conventional breeding, and transgenic approach. Fertilization is useful for small-scale production, whereas conventional breeding, and especially the transgenic approach, is the most successful form of biofortification for large-scale production. A study was done on rice grain to determine zinc concentration as affected by genotype, native soil zinc availability, and zinc fertilization. It had been suggested that rice with higher zinc concentration could be a way to improve zinc deficiencies in human populations that have a staple food of rice. Therefore, the study was conducted to evaluate the status of zinc in the native soil, as well as fertilizer application on zinc concentration in grains of 5 rice genotypes that were identified as being either high or low in grain zinc. These 5 different genotypes were grown at 4 different sites during the rainy season, where the native soil ranged in severely deficient zinc concentrations to extremely efficient zinc concentrations. 2 of the sites were located in the Philippines. During field preparations, the standard recommended dose of NPK was applied to all plots in order to ensure that the rice grain was not lacking in this nutritional area. The plots then received 15 kilograms of zinc, which is much larger than the recommended dose of 5 to 10 kilograms of zinc. The results showed that each location differed in the native soil concentration. There was no statistically significance in the average zinc concentration with application of fertilizers, and genotype differences in grain zinc concentrations were significant in all with a severely high zinc deficient soil. Also, where native soil zinc concentrations were highest, genotype zinc concentrations were at its maximum. In the end, they found that the dominant factor determining zinc concentration in the grain was the native soil, followed by genotype and fertilizer. Native soil being the most significant determining factor and fertilizer being the less significant factor. There continues to be research performed on this subject by organizations such as Harvest Plus, are determined to research further the benefits and complications of biofortification. Due to the need for more research on this subject, there has not been a solid conclusion on whether or not biofortification is the best way to decrease malnutrition in poor communities and countries across the world. We hope to find an answer soon.